This video covers Chapter 3, Sections 3.1 and 3.2 on amino acids, peptides, and proteins. The learning goals for Chapter 3 are to learn the structure and naming of amino acids, the structure and properties of peptides, the ionization behavior of amino acids and peptides, and methods to characterize peptides and proteins. Proteins are the main agents of biological function. They can serve as catalysts, such as DNA polymerase in DNA replication. They are important to transport, such as hemoglobin, which transports oxygen in the blood. They are important to structure, such as collagen in our connective tissue. And they are also important to motion, such as myosin, which is muscle tissue. The protein keratin is formed by all vertebrates, and it's the chief structural component of hair, scales, horn, wool, nails, and feathers. This is an image of the black rhinoceros, and this horn is composed of keratin. The black rhinoceros is nearing extinction because of the belief in some parts of the world that a powdered form of its horn has aphrodisiac properties. In reality, its horn has properties that are no different from human fingernails. Amino acids are important because they are the building blocks of proteins. Proteins are linear heteropolymers of alpha amino acids. And amino acids have properties that are well suited to carry out a variety of biological functions because they can polymerize, they have useful acid-base properties, they have varied physical properties, and varied chemical functionality. This is the general structure of an amino acid. The C-alpha carbon is connected to an amino group and a carboxyl group, a hydrogen, and then an R group. The nature of the R group will vary, and that will define what type of amino acid it is. The alpha carbon always has 4 substituents and is tetrahedral. And all of the amino acids except for proline have an acidic carboxyl group connected to the alpha carbon, a basic amino group connected to the alpha carbon, and an alpha hydrogen. The 4th substituent, the R group, is unique in glycine, which is the simplest amino acid. 4th substituent is just hydrogen. All of the amino acids are chiral except for glycine. So the alpha carbon, shown here, has 4 different groups attached to it, which makes it a stereocenter, a chiral carbon. And if you recall from organic chemistry, if something's chiral, that means it rotates plane polarized light in either a clockwise or counterclockwise direction. Now in organic chemistry, we taught you to use the R and S designation for a chiral carbon, but in biochemistry, we use the L and D designation. So to figure out if an amino acid is in the L form or the D form, you want to draw it like this, with the carboxylate group at the top. And then you ask yourself whether the amino group, the NH3 group, is on the left or on the right. If the amino group's on the left, then it's the L form. If the amino group's on the right, it is the D form. I could also convert this to the standard Fisher projection form, which looks like this. One of the great mysteries is that proteins only contain L amino acids. They don't contain D amino acids. So a question for you to ponder is, why did life evolve to only use L amino acids and not D amino acids? Now note that the relationship between these two molecules is an enantiomeric one. So they are mirror images of each other. They are enantiomers. Also notice that the solid wedges here indicate atoms coming out of the paper towards you, and the hatched wedges indicate groups going away from you. The amino acids are all classified based on their R substituents, and they can be placed in 5 groups depending on what that R substituent is: nonpolar, aliphatic, aromatic, polar uncharged, positively charged, or negatively charged. This is an overview of all of the amino acids divided into their particular subgroup based on the type of R group that they have. If you count up all these different amino acids, you'll get to the number 20. The universal genetic code, which is nearly identical in all life forms, only specifies these 20 amino acids. Nevertheless, many other amino acids are components of certain proteins. However, these other unusual amino acids result from the modification of an amino acid after it has been synthesized, with the one exception of selenocysteine, which involves a different process. And in fact, selenocysteine, not shown here, is often referred to as the 21st amino acid. This slide shows the nonpolar aliphatic R groups. So with glycine, you just have a hydrogen. With alanine, you have a methyl group. Proline is the only amino acid that contains this R group in a ring form. Notice that forms a 5-membered ring. Valine has this isopropyl group. Leucine has this 4-carbon isobutyl group. Here, isoleucine also has a 4-carbon group known as sec-butyl. And then methionine has a group containing a sulfide bond. 3 of the amino acids have aromatic groups. So the tyrosine and the phenylalanine have benzene groups. Tyrosine has an OH group here, which makes it different from phenylalanine, which does not. And then tryptophan has this aromatic group, which is called an indole ring, which is a type of heteroaromatic group. Hetero meaning you have an atom, the nitrogen, other than carbon in the aromatic ring. The groups with polar uncharged R groups are shown here. So serine has an OH group, as does threonine. Cysteine has a thiol group, an SH bond. Asparagine has an amide, as does glutamine, but they just differ in the number of CH2 groups between the alpha carbon and the amide. These amino acid side chains can form hydrogen bonds, and cysteine can form disulfide bonds. This slide shows the amino acids with positively charged R groups: lysine, arginine, and histidine. Now you may notice here that the way this is drawn, it's not charged, but at physiological pH there is a small but significant fraction of histidine that is charged and protonated, and that nitrogen here would be the nitrogen that would be protonated. Aspartate and glutamate make up the negatively charged R group category. The only difference between these two is the number of CH2 groups in that side chain. Amino acids contain at least 2 ionizable protons, each with its own pKa. The carboxylic acid has an acidic pKa and will be protonated at a low pH, whereas the amino group has a higher pKa and it will be protonated until you get to a high enough pH to deprotonate it. This shows the ionization of the amino acid glycine as compared with acetic acid. So notice here that the pKa of that carboxylic acid group, so that hydrogen, has a pKa of 2.34, which is lower than the carboxylic acid group for acetic acid. Why would the amino acid have a lower pKa? Well, that's due to the neighboring effect of that NH3 plus group, which is positive, and it's pulling electron density inductively away from the carboxylic acid, which shows how chemical structure can affect pKa values. Now if we look at the amino group, the pKa for that amino group is 9.6, which is lower than just a simple methylamine group. And the reason for that is similar. You have the carboxyl group, which is also electronegative and withdrawing electrons from the amino group inductively, thus lowering its pKa. The form that the amino acid will exist in, positively charged or negatively charged, will depend on the pH. At a low pH, the amino acid will exist in a positively charged form. At a high pH, the amino acid will exist in a negatively charged form. Now, between the pKa for each group, the amino acid exists in what's called the Zwitter ion form. That's where you have both a positive and a negative charge on the molecule. Here is the titration curve of glycine. and the ionic species predominating at key points in the titration curve are shown above the graph. The shaded boxes indicate the regions of greatest buffering power. So here we have on the x-axis the number of hydroxide equivalents that have been added, and on the y-axis we have the pH. So if we take glycine, the most acidic proton is the hydrogen of the carboxyl group. So when we add the base hydroxide, that will be the first one to be removed. So when we add that, the hydrogen is removed and we get to the carboxylate. Now this value here, that's where the pH equals the pKa, and this shaded blue region shows where there is a buffering capacity. After you remove the first acidic proton, you get to this form of the amino acid, which is the Zwitter ion, because the carboxyl group is negatively charged and the amino group is positively charged. And once you add one equivalent of hydroxide, you get to this point in the curve. That's where you have the Zwitter ion, and this, the isoelectronic point, which we'll talk about in the next slide, is 5.97. That's the pH at which an amino acid overall is neutral, or in other words, has a net charge of 0. Now when you add more than one equivalent of hydroxide, then the next most acidic hydrogen on the amino group will start to be plucked off by the hydroxide base to get you to this form. And again, you have a buffering region here, and this value is where the pH equals the pK2, and that's 9.6. So when you have two ionizable groups, so you have the COOH group and the amino group, in the titration curve you have two buffering regions. And whenever a molecule has more than one acidic proton, we can't just say the pKa, we have to indicate which one we're talking about. So that's why we use pK1 and pK2 here. The isoelectronic point is the pH at which there is a net charge of 0 for an amino acid, and Zwitter ions predominate pH values between the pKa values of the amino and carboxyl groups. So for an amino acid without an ionizable side chain, you can easily calculate this by simply summing the pK values for each ionizable group, the amino group and the carboxylic acid group, and divide by 2. So at this point, the net charge is 0. And at that point, it would not migrate in an electric field, and it would be the least soluble in water. And as I mentioned with the titration curve, amino acids actually have two buffering ranges. And this again is just pointing out those two buffering regions for glycine. Ionizable side chains influence the isoelectronic point of the amino acid. They can also be titrated, and the titration curves, however, get more complex. So this shows the more complex titration curve for histidine. So histidine has 3 ionizable groups because the side chain also is ionizable. It has an acidic proton. And above here we see all the different forms of histidine. So at low pH, histidine would exist like this with an overall +2 charge. So you have the side chain protonated, the amino group is protonated, and the carboxyl is in its uncharged form. So when you add some hydroxide, so you're adding a strong base, you're going to deprotonate the most acidic proton first, which would be the carboxyl proton. So you're going to remove this to get to this next structure. So as you add hydroxide, you form this second structure. And when the pH is 1.82, that equals the pK1. That's the pKa for the carboxylic acid hydrogen. And you would have a buffering region in this area. Now, if you continue to add more hydroxide, you eventually deprotonate all of the carboxylic acid, and then you start to deprotonate the next most acidic proton, which would be this one, the one attached to the nitrogen in the ring. So as you start to deprotonate that, you get to this structure, and when the pH is 6, that is equal to the pKr, where R is the abbreviation for the side chain. And so there would also be a buffering region here. And then if you keep adding more hydroxide, then you remove the final acidic proton, which would be the one on the amino group, to get to this structure. And here at a pH of 9.17, that equals the pK2, which is the pKa value for the hydrogen on that amino group. Now, where is the isoelectronic point? Again, that's the pH where the molecule has no net charge, and that would be where it's existing as a Zwitter ion. So this is the molecule that has no net charge. It has the carboxylate and the amino group, but they cancel out to give no net charge. And at a pi of 7.59, that means that the pH at 7.59 is when this amino acid has no net charge. Also in this curve, you would have a third buffering region. So there are 3 buffering regions for histidine. These tables are very useful. So it lists all of the amino acids, the molecular weight, and it shows the pK values for the carboxyl group and the amino group. And if there is an acidic hydrogen on the R group, it also gives the pK value for that, and it gives the pi value. And these values can be shown for all of the 20 amino acids, often called the common amino acids. So for lysine, for example, the carboxylic acid has a pK1 of 2.18. The amino group has a pK2 of 8.95, and then the R group has a pK value of 10.53. And then the isoelectronic point is 9.74. That's the pH at which there's no net charge. So if you didn't have the table where all the PIs are listed, how could you calculate the PI of a side chain? Well, you would write out all of the species that can exist. So you would draw all of the equilibria, and you would find the one where there's no net charge, which would be the Zwitter ion. So in this case, that's this compound. And then you add together the pK values on the left and the right of it, divide by 2, and that's the PI of that particular amino acid. Amino acids polymerize to form what we call peptides. Peptides are small condensation products of amino acids. So you take a carboxylic acid group and an amino group, and you form an amide. So amide is the functional group that you're forming. In biochemistry, we call those peptides. In this process, the OH and the H are lost, so water is lost, which is why we call it a condensation reaction. When we join 3 amino acids together, we sometimes call that a tripeptide. If we join 4 together, a tetrapeptide, etc. When a few amino acids are joined, the structure is called an oligopeptide. When many are joined, the structure is called a polypeptide. Proteins may have thousands of amino acid residues. Incidentally, when you form the peptide, the amino acids that make up that peptide get the term residue. The terms polypeptide and protein may be used interchangeably, but often when people refer to polypeptides, they're referring to things that have molecular weights below 10,000 Daltons. Peptide ends are not the same, so the amino terminal end is on the left, and the carboxyl terminal end is always shown on the right. And then the numbering and naming starts from the amino terminus. So if this is your example peptide, your amino acid 1 would be this amino acid. And the shaded area shows the amide bond, also known as the peptide bond. That's where the 2 amino acids were joined together via a condensation reaction. and the groups shown in red, those are the R groups for each amino acid. So that's how you can tell what amino acid was joined together. So you could test yourself and see if you can figure out what the different amino acids are in this peptide. When we name peptides, we want to start at the N terminus, and there are a few ways to do this. You could use the full amino acid names where you take off the I and E and add a YL, except for the last one. You could use the 3-letter code abbreviation, so each amino acid has a 3-letter code and a simple 1-letter code. This method is, I think, easier to use and look at than writing out the full name. Peptides serve a variety of function. They are hormones and pheromones such as insulin, oxytocin, and the fruit fly sex peptide. They can act as neuropeptides, antibiotics. They are found as toxins in things like mushrooms and cone snails. There are the common amino acids, but there are also uncommon amino acids that are found in proteins. They typically arise by post-translational modifications of proteins. So the proteins are made, and then after that, modifications are made to the amino acids. And there are also reversible modifications that can be important in regulation and signaling. Here are some examples of modified amino acids. So for 4-hydroxyproline, this group has been added at the 4 position. 5-hydroxylysine, we've added an OH group, a hydroxy group. 6-N-methylysine, we've added a methyl group. So the name tells you how it's been altered. Gamma-carboxyglutamate, a carboxyl group has been added, and this ends up being important in blood coagulation. Desmosine is formed from 4 different lysine amino acids. It is found in elastin, which is a protein found in connective tissue such as skin, lungs, and elastic arteries. Selenocysteine has a selenium atom, and pyrrolysine has a pyrrole derivative. We can also get reversible modifications of amino acids. Phosphorylation is an example of that, where we're adding these phosphate groups, or adenosine monophosphate could be added. Other modifications include adding an acetyl group or a methyl group, converting the carboxylic acid to an ester, or adding a methyl group here. Proteins vary greatly by size and number of residues. This table gives you some examples. So cytochrome C has 104 residues, but then at the other extreme we have titin, which has over 26,000 residues. However, sometimes very small peptides can have biological activity. For example, this is a modified peptide made from aspartate and phenylalanine, and it's been modified to form the methyl ester. And this we also know as aspartame, which is an artificial sweetener. So for the peptide to have some function in the body, it doesn't have to be big. Finally, proteins often have what's called a prosthetic group, which is a group that is covalently bound to it. Lipoproteins is a class of proteins where the prosthetic group would be lipid. Glycoproteins are covalently bound to carbohydrates. Phosphoproteins are covalently bound to phosphate groups. Hemoproteins are bound to heme. Flavoproteins are bound to flavin nucleotides. And metalloproteins are bound to a variety of metals. So in summary, this video has discussed the variety of amino acids, how they ionize at different pH, the isoelectronic point, the chirality of amino acids, the fact that amino acids have buffering regions, and that they can be combined in great variety to form a huge number of different types of proteins.